Can somebody confirm you can hear me?
I can hear you.
Thank you. All right, before we begin, are there any questions? All right, let's begin. Today is January 28th, and we are going to be covering Lab 5, Streaking for Isolation and Culture Media. You also should be beginning to read the unknown project instruction files. You can find it on Canvas. You need to have this read by next week because I'm going to discuss and we're going to start the unknown project next week. Boy, I'm not ready for doing that. I'll have to get ready this weekend. All right, let me show you where that unknown project instruction file is. Under week 5 of the lab. The network is still slow at Clark today. Week 5 lab. Read your unknown instruction files right here under week 5 lab. All right, are there any questions before we begin the lab? If not, lab 5, cultivation of bacteria, culture media, inoculation, and isolation. You need to do some reading. Today it's not very much. Well, maybe 3 pages, 4 pages about culture media, and then about 1 page on obtaining pure cultures. There is some video clips in today's lab. We no longer have a virtual lab because the one we had requires Flash to run. And Flash is no longer supported and will not run on people's computers. The primary learning objectives upon successful completion of this lab, you should be able to, 1, understand the importance of proper specimen collection and transport to the lab. 2, explain the 5 principal steps used by microbiologists to process specimens. 3, differentiate between various types of culture media. 4, explain the purpose of streak plate culture technique, aka streaking for colony isolation. And then 5, define the following terms: what's selective media, differential media, and what is reducing media. Any question about any of that? All right, the ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, meaning they'll dry out, and others are sensitive to the exposure to oxygen. Most anaerobes are sensitive to the exposure to oxygen and therefore require a special transport medium to keep the cells alive. Once a specimen is received in the lab for microbial culture, microbiologists employ the 5 principal I steps to process it. First, inoculation; second, isolation; third, incubation; fourth, inspection; and fifth, identification. So 1, inoculation, that's placing the specimen on an artificial culture medium to initiate growth. Isolation, you then need to spatially separate the microbial growth into distinct isolated colonies. 3, incubation, that's placing the culture medium in an environment having conditions to facilitate the growth of microbes. 4, inspection, examining the culture for the presence of microbial growth. And then 5, identification, you normally have to perform specific tests to identify any pathogenic microbes that may be isolated. Any question about any of that? All right, First, we need to discuss the different types of culture medium. You're already familiar with general purpose media. We discussed nutrient agar or Na. We discussed nutrient agar with yeast extract or Na, and we discussed TSA or tryptic soy agar. All of these are rich media that supply many nutrients, allowing many organisms to grow. However, fastidious organisms or fastidious microbes require very specific nutritional requirements that must be added to the media in order for them to grow. So a fastidious species will not grow on any general purpose media agar plate. Any question about any of that? So to get a fastidious species to grow, you must grow it on an enriched general purpose media. One enriched general purpose media is where we add 5% sheep's red blood cells to TSA to get the growth of a species like Streptococcus pyogenes, which will not grow on a general purpose media, but it will grow on blood agar because the red blood cells supply the nutrients required for this species, which in this case is Streptococcus pyogenes, to grow. Any question about any of that? All right. So there's general purpose media and obviously, enriched general purpose media. And then there's selective media. Selective media is a media that contains chemicals that inhibit or halt the growth of some organisms, but do not inhibit the growth of other organisms. Keep in mind two things when using a selective media. First, a selective media isn't necessarily perfectly selective. Given enough time, an organism which is selected against in a selective media can eventually begin to grow. And then second, even those organisms which are supposed to grow in selective media may grow slower in selective media or form smaller colonies in selective media than they would in a general purpose media. Any question about any of that? I should give an example. An example of a selective media is McConkey's agar. It contains chemicals or additives that prevent the growth of gram positives, and it has, so the gram negatives are selected for. We'll talk about McConkey's in a little bit later, just a little bit. All right, we also have a differential media, and this is a media that contains chemicals that produce visible differences among different groups of organisms when grown on that same medium. These differences are unrelated to how well the organisms can grow on the medium. Differential culture media allows you to see dissimilar bacteria that are visually distinguished from each other by the differential media. So when you're growing two species on a differential media, they may have different colors, for example, when they grow. And then lastly, the last media we're going to talk about, at least in this lab, is a reducing media. This media has chemicals in it to promote the growth of anaerobic bacteria because low or no oxygen conditions are required to culture these organisms. Usually a reducing media has a chemical that combines with oxygen, removing oxygen from the media. All right, any question about these different types of media? It's important to note the culture media can be classified into more than one category. For example, McConkey's agar is both selective and differential. Blood agar is both an enriched media, enriched general purpose media, as well as a differential media. McConkey's agar is both selective and a differential. It is selective because it has additives that allow gram negatives to grow, but select against gram positives. So usually if we get growth on a McConkey's media plate shown here, it will select against gram positives, they usually don't grow, and select for gram negatives, they usually do grow. So it is selective. It's also differential on the ability of the species to ferment the sugar lactose. If the bacteria can ferment the sugar lactose, it will change the pH because lactose fermentation will produce acidic products, changing the color of the plate to this dark pink or purple color. And the growth of the colonies will be the same color, purple or pink, dark pink. And the plate around the colonies will also be purple or dark pink, probably purple. That's more like dark pink. If, on the other hand, the species can grow on the plate, it'll be most likely gram negative. But if it cannot ferment the sugar lactose, then the color of the species will be its normal color, which in this case is white or off-white. No fermentation of the sugar lactose, fermentation of the sugar lactose. And then both of these are gram negative because they're growing on McConkeys. Any question about that? Blood agar is both an enriched general purpose media and a differential plate. You can grow a species on the blood agar that cannot grow on general purpose media. One such species is Streptococcus pyogenes, the bacteria that causes strep throat. It can grow on a blood agar plate because the blood supply the nutrients that this fastidious organism needs to grow. It's also differential because you can differentiate between the different hemolytic patterns of different species growing on the plate. You can have beta hemolysis, shown here. The B or the beta is where the bacteria are growing, and that is really a white growth there where the beta is. And then the clear region around the beta and inside the beta is where all of the bacteria have lysed the red blood cells. So all of the red blood cells in the clear region are lysed. And you can actually see through this region of the plate. I like to hold it up to either look at the clock or look at the light bulb in the light. So you can see through the clear region of the plate. You can't see through the beta because that's the bacteria. You can't see through the red region either. Anyways, that's beta hemolysis. This is alpha hemolysis, where different species of bacteria, shown in the off-white alpha, is growing on the plate. And if we look at the red of the blood, it's red far away from the growth, but right near the growth, we have a darkening, a dark ring around the alpha. And inside the alpha, it's totally a dark ring. And this is showing you what alpha hemolysis looks like. It either is a darkening or a greenish ring around the alpha. And that's alpha hemolysis. Alpha hemolysis is the partial lysis of the red blood cells. So some of the red blood cells have been lysed, causing the darkening color or the greenish color. And then out here, where it's all red, there's no lysis of the red blood cells. The third hemolytic pattern is gamma hemolysis, which is no hemolysis. You can see the red of the plate come all the way up to the white growth of the bacteria. And if you don't know, that's alpha, beta, gamma, the three first letters of the Greek alphabet, which is alpha hemolysis, beta hemolysis, and gamma hemolysis. Any question about any of that? Let me shrink that down. All right, this table is showing you the different types of media. The general purpose media, we have three: TSA or tryptic soy agar, Na or nutrient agar, Na or nutrient agar with yeast extract. We also have a differential and enriched medium, which is both differential and enriched, and that is blood agar or BAP, blood agar plate for BAP. We also have an enriched general purpose medium, which is chocolate agar or CAP. Note that chocolate agar is not differential, it's only enriched. And then we have selective and differential media. All of McConkey's or MAC, eosine, methylene blue or EMB, or hectone, agar, or HE. All of these are both selective and differential. They are selective in that all three of them allow gram-negative cells to grow, but select against gram-positive cells. And all three of them are differential on the fermentation of the sugar lactose. McConkey's agar, if it can ferment the sugar lactose, and if it grows on the plate, the bacteria will appear dark pink or purplish. EMB, or eosine methylene B, if the bacteria can grow on the plate, meaning they're gram-negative, and if they can ferment the sugar lactose, then the bacteria will either look metallic green or a very dark, almost a black color. And then if the bacteria are growing on HE or hectone agar, if it grows on the plate, that means it's gram-negative growth because the gram-positive is selected against. And it's differential on the fermentation of the sugar lactose. If the bacteria can ferment the sugar lactose, then the bacteria in the region around the plate will be a salmon color, that is a yellow-orange color. And if it cannot ferment the sugar lactose, the bacteria will be its normal color, which is usually off-white or white, but it could be some other color as well. And then we have a selective medium. PEA is the only media we use, which is only selective. PEA has phenyl ethyl alcohol in it, what's called PEA or phenyl ethyl alcohol medium, which selects against gram-negatives. PEA gets on the cell wall of gram-negatives and then prevents the cell from growing. So it selects against gram-negatives and therefore selects for gram-positives. And then we have mannitol salt agar, which is both selective and a differential media. It is selective because it has 7% sodium chloride in it, and only organisms which, like salt, can grow on this plate. Most bacteria will not grow above 2% sodium chloride, and this plate has 7% sodium chloride. So something that, like salt, can grow on mannitol salt agar. Something that doesn't like a fairly high salt concentration will not grow on mannitol salt agar. It's also a differential medium plate because you can differentiate between the fermentation of the sugar mannitol and the species which cannot ferment that sugar mannitol. If the species can grow on the plate, it tolerates salt, and if it can ferment the sugar mannitol, the growth will be yellow, especially a yellow halo around the colony. If, on the other hand, it can grow on the plate, that means it tolerates salt, likes to be in a high salt environment. But if it's not yellow, it cannot ferment the sugar mannitol. It does say you can use this plate to separate Staphylococcus aureus from other species of Staphylococcus. Because Staphylococcus species can grow on the plate, they like salt. And then Staphylococcus aureus will be yellow, and at least most of the other Staphylococcus species do not ferment the sugar mannitol, so they will not turn yellow. Let me warn you that other species besides Staphylococcus can grow on a mannitol salt agar plate. Any species that tolerates high salt can grow on a mannitol salt agar. For example, Micrococcus luteus, a bacteria that can grow on your skin, can grow on mannitol salt agar. And Micrococcus luteus does not belong to the family Staphylococcus. Any question about any of that? All right, here we have inoculation where we collected things falling out of the air on this plate and then incubated it. And so this is the culture of microbes obtained from the air. Then next we have isolation. Louis Pasteur first developed the concept of working with pure cultures. He also developed a way to obtain a pure culture. If you don't work on a pure culture and then your test result is, let's say, positive, you don't know which species in the mixed culture caused the positive result. So usually microbiologists work with a pure species. How Louis Pasteur got his pure species was he took his liquid culture of bacteria and made several different dilutions of that bacteria. And then each dilution, he replica plated it, meaning he grew each dilution in 10 separate media tubes. And if one dilution only had one tube that had growth in it, Louis Pasteur correctly concluded that the growth was likely to be a pure culture because one or a few cells started the growth in that tube. So you have to have one tube with growth and 9 tubes with no growth. And then Louis Pasteur used the growth in that one tube to obtain his pure culture. It was a very difficult way and time-consuming way to obtain a pure culture, but it did work at least most of the time. It did not work all of the time. And to tell you the truth, I've never isolated a culture that way, so I'm not sure uh, how reliable it was. It's obviously not 100% reliable. And I don't even know if it's 90% or higher reliable, meaning 90% of the time you got a pure culture. I don't know what it would be. It's probably a little higher than 70%, but it may be only 70%. I don't know. Like I said, I've never done it, so I don't know. We have Dr. Koch, who invented an easier way of obtaining a pure culture. What Dr. Koch did was he took out his culture and then streaked it on a plate of solid media to obtain isolated colonies. And then he took from one isolated colony, streaked it out on a second plate to once again get isolated colonies. And if he took cells, or cells from one colony on the second plate, if you took cells that went through 2 colony isolation events, then there's a very high chance, it's greater than 95%, that you will have a pure culture. It's not 100%, but 95% or higher is good enough in microbiology, and this is the procedure we use to obtain a pure culture. You need to streak it out for colony isolation, streak those cells out 2 times, going through 2 separate isolated colonies. And then you can say with 95% certainty or higher that you have a pure culture. All right, any question about that? Let's talk a little bit more about streaking for isolated colonies, usually called streaking for isolated colonies or colony isolation. What you do is you take your loop, sterilize it. So you either put it in the flame or you put it in the back to incinerator till it's red hot. Then you cool it, pick up the cells you want, and then streak it on sector one of the plate. And it actually looks like this one. They started here and then worked that way, which is very weird. And then you sterilize your loop to remove any cells on it, and then cool it, and then you begin a streak in quadrant two. This isn't quite done correctly because um, you should start the streak outside sector one and then go into sector one 3 or 4 times. So they got that more like 6 times. You should only do it 3 or 4 times. And then the other problem is you should go in sector one where the bacteria are diluted. So right here, the bacteria are very high in concentration. So that's the first streak, and that's the second streak, very high. Third streak, little lower. Fourth streak, lower. Fifth streak will be the lowest. So you should only be going really in the last 2 streaks to pick up the cells for quadrant two. And you want to go into quadrant one 3 or 4 times. So there's 1, 2, 3 times. And like I said, they did it 5 times. You should really only do it 3 or 4 times. So 1, 2, 3, 4 would be perfect. And then don't go where in streak one, in where the bacteria are more concentrated, go in the regions where the bacteria are more dilute. You don't want to pick up a high concentration of cells, otherwise streak two will look very similar to streak one. And then streak three is better. They started over here. What's the problem with streak three is, once again, they went where the bacteria are very high and concentrated. You only want to go where the bacteria are more dilute. And you want to only do that 3 or 4 times. So you should stop right about there. Shouldn't come back here. And then streak it out. And then actually, streak four is done correctly. You start where there's no cells, go through streak three to pick up cells. And then you do it 3 or 4 times. So I guess they did it more like 6 times. Do it only 3 or 4 times. And then in each sector, it's not shown here, you should be streaking it out between 20 and 30 times. So this is 1, 2, 3, 4, 5, 6 times. You should be streaking it in one sector 20 or 30 times. And then streak it out here. This is pretty good in sector four or quadrant four. And you'll notice that in quadrant four, we're getting isolated colonies. Now remember, to get a pure culture, you take from one isolated colony here, streak it out on a second plate for isolated colonies, and then pick an isolated colony, take the cells from one isolated colony to set up your stock culture. And this will be 95% chance or higher that it'll be a pure culture because it's gone through 2 colony isolation events. And most colonies are formed by one cell or a small number of cells that were isolated from one cell, but not all of them. Like that colony is much bigger. That colony is much bigger than some of the other colonies. These were definitely formed by 2 or more cells. And as you can notice, about 30% of the colonies here will be formed by more than one cell. So if you only streak it out one time, your chance of getting a pure culture is around 70%. And that's not good enough for microbiology. You need to streak it out 2 times and then take cells that went through 2 colony isolation events. And then your chance of getting a pure culture will be 95% higher or greater. And that is good enough for microbiology. If you don't know, you can never be certain that you obtained a pure culture to 100%. And that's just the facts of life and biology. And that at each time you streak it out, it's possible that you could get that colony formed by two or more cells. I actually have a friend, and he's another instructor who teaches microbiology at Clark, who was working with a species of bacteria, and there were nitrogen fixing bacteria, and he did not supply nitrogen in the media. And it turned out that he needed to have two species of bacteria growing together, or else he wouldn't get any growth because the bacteria needed nitrogen. And for some reason, I don't remember the specifics, you needed both species there to convert or pull down nitrogen from the air and then get it so that both species could use it. And so he was streaking out these colonies, and he thought he had a pure culture, but it turned out it was actually formed by two different species. But that's very rare. For the first place, we don't use media that the media we use normally supplies nitrogen. We'll just word it that way. All right, any question about streaking for colony isolation? All right, our next step of the 5 steps is 3, incubation. Recall from lab 1, the culture media contain chemical nutrients necessary for the microbes to grow, but additionally, you must provide an appropriate physical environment to culture microbes. In our lab in Clark College, we have simple incubators where we put the plates of our microbes to grow, and all it does is it warms up the plate to a certain temperature, usually 30 degrees Fahrenheit or 37 degrees Fahrenheit, but you could set the temperature at a different temperature. And the incubator has a pan in it that we put water in, so it does supply humidity. That's important because if you don't have a pan of water providing humidity, the plates will dry out in about 3 days. And so you will only have growth for 3 days. After that, the plates will dry out and the bacteria will die. But if you put the pan of water in there and have humidity in the air in your incubator, the plates can go for at least a week. Sometimes they can even go for 2 weeks. And that's important. Anyways, those are our simple incubators. They don't have all these Dobbs and Niles on it. But you can have a more complex incubator. This one here has these Dobbs and Niles to control how much oxygen is in the incubator to expose the cells to. And then the other Dobbs and knobs are to control how much CO2 is in the incubator and how much CO2 is being exposed to the bacteria. Like you would want an incubator for that. For growing something like Neisseria gonorrhea, it prefers growing at reduced oxygen conditions. It doesn't like about the 20% oxygen in the air. So you need to grow it at less oxygen, something like between 4 and 10%. And you can do that in a complex incubator. I think it likes higher than CO2 as well. So usually you put more CO2 in to grow that. But we don't have any complex incubators like this in our lab. So oxygen, the amount of oxygen, the amount of carbon dioxide available, the temperature, the humidity, I talked about all of those. And then the time, that is when you put the cells or culture into the incubator, and then when you pull it out. If you have a real slow growing organism, you can grow it in the incubator for longer than one class to the next. All right, any question about 3, incubation? And then 4, you can do inspection. There are obvious visible colony morphological differences between different species of bacteria. Microbiologists can describe the appearance of the colonies by a variety of descriptors: size, shape, margin, elevation, color. Let's come on up here. So you can see that species is definitely different than that species, even though they're both yellow. That's a very dark yellow, that's a light yellow. It's different than that species, different than that species. These are the same color, but this one has a different margin than this one. Any question about that? Usually, just by looking here, you cannot determine what species this one is, what species that one is, what species this one is, or that one. Sorry, that one there. You can't do that until you ran additional tests. So the next is identification, where you run specific tests to determine what species you have growing. And you have to do the biochemical testing. We'll talk about that in later labs to determine what species you have. All right, know the terms of this lab. Don't need to look at the references. Everything we do will be done online, but you do need to understand what this lab is discussing, what you're answering in the questions. You also need to view the videos to see what's going on in the videos. In the first video we have a video on inoculation of an auger plate using streak plate culture technique, meaning we're streaking for colony isolation. In this video, you only need to watch it up until 4 minutes and 15 seconds. The video is also only streaking 3 sectors, and we showed you 4 sectors above. It doesn't matter as long as you get isolated colonies. Oh, let me state one other thing on getting isolated colonies. It doesn't matter where you get the isolated colonies. Here we're getting it in quadrant 4, but if you had a more dilute culture of bacteria to begin with, you might get your isolated colonies in quadrant 3. Or if it's even more dilute than that, you could get your isolated colonies in sector 2 or quadrant 2. And if it's a very dilute culture of microbes that you're starting with, you might even get your isolated colonies on sector 1 or quadrant 1. Any question about any of that? All right, we have 3 little videos on beta hemolysis, showing you beta hemolysis, alpha hemolysis, and gamma hemolysis. These are all short little videos. We also have a video talking about McConkey's auger. In this video, you only need to watch up until 3 minutes and 50 seconds. After that, you can watch it, no problem, but you must watch it for 3 minutes and 50 seconds and understand what's going on. We have a little video on mannitol salt auger as well. Go ahead and watch that video. We used to have a virtual lab where you could practice streaking your plate. Unfortunately, we don't have that anymore. And then answer the questions in the worksheet. 1, describe a natural circumstance where Staphylococcus species of bacteria might benefit from salt tolerance, meaning a natural place where Staphylococcus can grow in high salt concentration. Do not say a mannitol salt auger plate, because that is not a natural circumstance. 2, how might hemolysis benefit a hemolytic organism? So we have a bacteria and it's infected the patient, lysing their red blood cells. What benefit to the bacteria can come from lysing the red blood cells? 3, imagine that you have a culture containing a single bacterial species. This culture may be one of several possible species. Describe where you can use mannitol salt agar, blood agar, in EMB plates to help you identify the unknown bacterial species. That is, how would you use these plates to help you identify the species? And you must discuss two properties of each plate. I thought I had that in the worksheet. I'm not in the worksheet. Let me get to the worksheet. Discuss or name two properties to each of these media plates that help you identify the species. Any question about number 3? Number 4, what is the main goal of streaking for colony isolation? Number 5, what happens to the growth on the plate if you do everything correctly, but you forget to sterilize the loop in between sectors when streaking for isolation? In your answer, be specific, and you need to discuss the growth on each sector of the plate. Now, don't tell me there will be contamination because you're doing everything correctly, and it says you're not streaking between the sectors. That means you are streaking before you streak out sector 1. Any question about question number 5? 6, what happens to the growth on the plate if you do everything correctly, but you transfer from stock culture to inoculate each of these sectors? Be specific, discussing the growth you see in each sector. Normally, when you're streaking out a plate, you only take cells from the stock culture to set up sector 1. Well, in this question, what happens if you set up each sector from cells in the stock culture? 7, suppose you successfully streak a plate and obtain isolated colonies of 2 different bacterial species. That is, you streaked from a mixed culture. How can you use this plate to create a new pure culture of only one of the bacteria? Be specific in your answer. Any questions about number 7? All right, 8. In the table below are a variety of agar plate media available to microbiologists to cultivate bacteria. To note whether each medium is non-selective, selective, and/or a differential media by placing an X in the appropriate column or columns. You may use the table of media in the lab module to answer these questions. So nutrient agar, is it non-selective, selective, or differential, or any combination of the 3? I'll tell you off the start that you'll never have all 3 together because you cannot have a media that is both non-selective and selective at the same time. It'll be either one or the other. For TSA, do the same. For CAP, that's chocolate agar, do the same. For BAP or blood agar, do the same. McConkey's, EMB, and HE, do the same. Mark whether it's non-selective, selective, or differential. For MSA, do the same. And then for PEA, do the same. Any question about number 8? For number 9, which agar plate or plates were inoculated without using streak plate technique, meaning without streaking for colony isolation? Plate A, plate B, or plate C? Any questions on this lab? All right, it's 7:16. I will be here until, let's make it 7:45, or until the last student logs out. So if you have a question, make sure you ask before the last student is logged out. Oh, okay, get started on the lab. Like I said, I'll be here to help you. I'm a resource if you need me.